glad to have this opportunity to introduce you our scalable microfluidizer technology and talk about how you can use it for producing RNA delivery platforms. Just a brief agenda for my today's presentation. I'll give you a very brief introduction to RNA therapeutics and its delivery. I think everyone here knows better than me. And then I'd like to talk about a little bit some common challenges and requirements during the manufacturing of ROSE platforms, and then move on to introduce you our company, Microfluidics. And so my focus will be explain the technology and hopefully use some examples, although they won't be super scientific, but I do want to give you some examples to demonstrate the versatility of this system and how you can use it in producing different kind of delivery systems. I will end my presentation by showing you our diverse product portfolio, and then be happy to answer any questions. So, as I mentioned already, I think everyone knows this very well. I'll just by saying that the nucleic acid-based, including the RNA-based therapeutics, have demonstrated their great potentials, especially in the past few years, with increased number of approved therapies and vaccines. I think everyone also knows very well that the challenge remains as how you can deliver the nucleic acids effectively and efficiently. So many strategies have been extensively investigated. I think to me, the popular ones are generally fall into two different groups. One is the viral-based platforms, and the other one are non-viral-based nanoparticles, which maybe more people are interested. So I have a few examples here listed, which, by the way, are all good applications for the microfluidizer technology. So for the non-viral-based nanoparticles, generally there's different kind of lipid-based nanoparticles, there are polymer-based nanoparticles, and there's a few other different kind of nanoparticles. I've heard pretty novel particles this morning, so they all belong to the nanoparticle groups. And then there's viral vectors, and which utilizing several different kind of viruses to help with the delivery. So the system kind of evolved, and especially for the lipid-based nanoparticles to the lipid nanoparticles, which have made a pretty good breakthrough in the past couple of years with the pandemic situation, and I think a lot of people are interested in the lipid nanoparticles. So to us, we like to use a broader definition as a lipid-based nanoparticle, but I do think some people view lipid nanoparticles as a group of special particles that are formulated with specific lipid ingredients. And despite the success and the most interest for the lipid nanoparticles, the RNA lipid nanoparticle system still needs a lot of work, and they have their own limitations and issues. So one of the critical issue is the lack of stability, especially for the long-term stability. Despite all the effort trying to increase that, all currently authorized vaccines still require frozen storage with temperatures as low as minus 80 degrees Celsius. So this is where other lipid-based particles, or maybe even other nanoparticles, can help, because despite all the concerns or different views, they have been proved they work very well. They can also offer their own benefits. For example, cationic nanomotion. GSK has a platform with their cationic nanomotion platform that demonstrated their great results, working very well. And I learned there's a couple of nanostructured lipid carriers that has showed great efficacy in some late-stage clinical evaluations. So on the other side, I think most people may be more interested in the nanoparticle-based platforms, but I hope there are people working with viral vectors, and so let's not forget about them, because so the viral vectors by far have achieved, in my view, greater clinical successes with more approved products on the market, and also have a bigger pipeline, and well, maybe not bigger pipeline, but it's more coming in the pipeline. So regardless of which platform you choose to work with, there are many factors and key quality attributes you need to consider, starting from development all the way to manufacturing. So I have by no means to give you a complete list. I'm just going to talk a few of them which are relevant to our technology here. So first and foremost are physical chemical properties. Among many of the important factors, I would say particle size characterization is certainly one of them, especially to be able to achieve desired particle size and distribution, because the size and distribution will not only affect the stability of the system or the particles, but it can also affect both in vitro and in vivo performance. So once you optimize your process to achieve good size and distribution, you would want to reproduce those particles every time with batch-to-batch consistency. And the choice of your manufacturing or method should also be somewhat future-proof in terms of scalability. So the technology should be readily and easily scale up with guaranteed results. Some of the current methods to produce lipid nanoparticles or viral vectors involve using chemicals such as solvents and detergents. This is not only put additional burden to your downstream processes, because you have to have separate process to remove the residual chemicals. It's also facing a lot of pressures because solvents are not desired with the increased concern in health and safety. So the possibility to reduce or even eliminate the use of chemicals is a huge, great plus to the process. It's not only make it a simpler process, but also more environmentally friendly. One more point is it will be good that if your process can improve or help with downstream or adjacent processes. So for example, back to the particle size, the in vivo delivery requires sterilization of the product, and this is usually done through terminal sterilization using sterile filters. I think everyone knows very well that the filters use membranes with really pore size of 0.22 microns or 220 nanometers, which implies if you use this method, your majority of the particles should be less than that size, and to ensure an efficient filtration process. I've talked about the undesired to use chemicals potentially can save the downstream process such as TFF. So last but not least is the manufacturing equipment should allow the entire manufacturing process to comply with regulatory requirements such as cGMP. Many processes also require, I think CIP is a must, and some aseptic process would also require SIP for the process. So in a quick summary, Microfluidics technology can address all these challenges and requirements from development to production with our unique benefit to achieve target results and guarantee the scalability. Before I explain how the system works, I just want to quickly introduce Microfluidics as a company to you. So Microfluidics is part of Material Processing Technologies, or MPT platform, under the Health and Science segment of our parent company, IDEX. Microfluidics itself was founded almost 40 years ago. We specialize in design and produce high shear fluid processors using our core interaction chamber technology. We're located in Massachusetts, just outside of Boston, about 30 minutes from here in Westwood, if you know that place. We have a global presence with many units sold to customers all over the world. I already mentioned that we're part of the ADEX MPT platform. The Microfluidizer technology is used in many industries, but primarily in pharmaceutical and biotech industries. For example, we have many units are used in both development and commercial manufacturing of a lot of approved drugs and vaccines. But we're also widely used in several other industries. With our extensive experience in applications and machine designs, especially around cGMP compliance, we'll be able to tailor a solution to enable our customers to achieve your specific processing goals. So the technology and how the system works is illustrated in this diagram. So you can see from this corner here that product with large particles enters the inlet reservoir and is cooled into a constant pressure. pumping system which pushes the material through a fixed geometry interaction chamber at pressures up to 30,000 psi. So inside the interaction chamber, the material will experience consistent high shear forces and impact forces, and all the material get treated exactly the same way to achieve uniform results. If needed, temperature can be efficiently controlled afterwards. We have two different chamber designs. The one showed on the top there is the white type chamber. So the white chamber has two microchannels, and the product impinges on each other after passing through the chamber, which makes this design ideal for producing most of the nanoparticles systems, including nanoemulsions, liposomes, lipid nanoparticles, and polymer nanoparticles. The one shown at the bottom is our Z-type chamber. So the Z chamber only has one microchannel, and this design is ideal for processing any product that has solids in there for deagglomeration, particle size reduction, and this design is also good for cell disruptions. As I mentioned, one of our advantages is scaling up with our technology. So when we scale up, unlike some other conventional technologies, we don't increase the size of our chambers because that's basically ruining all the parameters you have been working with. Instead, we use multiple parallel aligned chambers, or microchannels, I should say. So material will receive, as long as you keep the process conditions the same, material will receive exact high shear and impact treatment inside the microchannels. That's help you achieve guaranteed scale-up results. And with that, lead to my first group of examples for the application in the lipid-based nanoparticles. So I think many people are familiar with the solvent-non-solvent mixing method to produce lipid nanoparticles. So this is essentially what we call the bottom-up method, and there's a few different ways, as illustrated there, to mix the solvent and non-solvent stream. Regardless of what kind of mixing method you're using, the bottom-up method actually created individual nanoparticles via controlled precipitation. So the key to the process to be successful is to precisely control the stream of each the flow rate of each stream and maintain their ratio. Whereas if you use our system, we're more with the conventional top-down approach, meaning you start with larger particles and then reduce the size down to your desired final size. And you can either choose a one-step in situ encapsulation with nucleic acid, which is similar to the bottom-up method, meaning you mix your lipids and your nucleic acids all together and you process through and you achieve encapsulation. Or you can choose something what we call the two-step approach, where you create blank particles first, then simply introduce or load the nucleic acids afterwards by complexation. And some people will call the particles obtained or the system obtained by this way lipoplexis. So although I think the exact structure of the RNA LNP system is still up for debate, most people believe that if you do in situ encapsulation, most of the nucleic acid will be located inside the nanoparticles. And if you do the two-step approach with the complexation afterwards, most nucleic acid are likely going to be around the surface, which also leads to some people think this may not offer as good protection to the nucleic acid as internally encapsulated. However, I've seen a lot of study, including actually one I'm going to show you in a minute, have confirmed that this is not the case. Even with the two-step approach with complexation afterwards, good protection and stability can be achieved by the particles. And plus, if you do this way, the two-step approach, you can get some additional potential benefits, such as you can achieve better stability, and some of the particles may offer some other properties, such as be used as adjuvant or stuff like that. So with that, I'd like to show you a quick proof of concept study we did with our collaborator at the University of Strathclyde. So here we tested a LNP formulation to encapsulate nucleic acid, and specifically it's a DNA, model DNA in this case, using both the one-step and two-step approach. what I like to point out here is in either approach, we did not use solvent. So in the process, we simply just mix all the lipid ingredients together, let them hydrate in the buffer, and process through the microfluidizer processor. You can see from here, with either approach, you can achieve good particles with good size and distribution, and also either cases achieved excellent DNA loading with integrity. So the idea of using the two-step approach to help achieve long-term stability is actually illustrated in this publication just published, I think, last month or this month, by researchers at Access to Advanced Health Institute, which was previously known as the Infectious Disease Research Institute, which is also a long-term partner of us. So in this study, AHI has developed a nanostructured lipid carrier, or NLC, to deliver their Replicon RNA vaccine candidates. So the efficacy of that, of the Replicon RNA and the NLC system has been demonstrated and extensively discussed in their previous publication, so which is now the focus of this study and my presentation. So here, I just want to show you the approach to help increase the stability. So the NLC was formulated with both liquid and solid lipids and also a cationic lipid. And the system was processed through our system, and you can see the formulation produces particle stable for at least 12 months, and they're either refrigerated condition or the room temperature condition, with their component concentration remained unchanged during this period of storage time. So the stability of the particles was slightly longer, as shown in this plot here. You can see the NLC was still able to complex with RNA even after storing for at least 21 months at the refrigerated condition. And despite we mentioned that most of the RNA was considered to be at the surface of the lipid, you can see from the graph here that the NLC was able to offer good protections against the RNA's degradation. So this is very promising, and AHI took a further step to investigate the feasibility of lyophilizing the entire vaccine formulation after the complexation, which you can see from here. The middle graph here shows the lyophilized sample. It was able to maintain an elegant white cake throughout the study, and can be reconstituted into a milky white solution just as freshly prepared complex. And they evaluated two model RNA. So the one showed on the top was a self-amplifying RNA. The one showed at the bottom was the mRNA. And so in either case, the integrity of the RNA was able to maintain, as shown on the with the size graph there. And the one in the middle up there showed the lyophilized vaccine sample was able to induce neutralizing antibody titer identical to the freshly prepared vaccine sample at the same dose. So the lyophilized vaccine samples also displayed excellent long-term stability. It's shown on the top left graph here. Under different storage conditions, even at elevated conditions of 40 degrees Celsius. And in most of the cases, as the graph shows here, the RNA, the integrity of the RNA was maintained after lyophilization or frozen conditions. And in certain conditions, you can see at the bottom, it's the bars are a little bit faded, but you can tell from some of them that after lyophilization, the NLC was still able to offer protection to the RNA against the RNA's degradation. Although some of them are starting to diminish, but when you evaluate the in vivo protein expressions as shown to the right there, some of the storage conditions are still able to yield high levels of protein expressions, especially like the one at the bottom, that the lyophilized sample stored at 4 degrees Celsius was able to induce expressions that with no significant difference compared to freshly prepared complexes. So this is, this study clearly demonstrated the proof concept of lyophilizing the RNA and NLC systems here, and indicated their great potential for long-term thermal stability. My last example here is trying to demonstrate the process scalability. So here, my example was actually using a similar adjuvant nanomotion formulation. So you can see here that's the particle produced from a pilot system, which is a M110EH unit. When the same formulation was processed with our production system, in this case with a 50 horsepower machine capable of pumping up to 5 liters per minute at 30,000 psi, you can see the results were almost identical, with the particle size overlapping on each other. In this particular case, with the optimized formulation, which is a more concentrated formulation, the scaled up capability can be done with just a 200-liter batch, which can produce up to 5 million doses per day, and the entire manufacturing process from sample preparation to post-process sterilization can be completed in less than 8 hours. With that, I'm going to switch topic and talk quickly about viral vectors, specifically the adeno-associated virus, AAV. So AAV is currently the leading platform for in vivo gene therapy delivery because it's non-pathogenic and it's episomal. With its many serotypes, the AAVs can also reach different target sites. Although the AAV has a limited packaging capacity due to their limited, relatively smaller size compared to other viruses, this is not an issue for RNA interference-based therapeutics because it's usually employ smaller sized expression cassettes. So how does AAV relevant to the microfluidizer technology? As briefly illustrated here, so the AAV vectors are commonly expressed in either mammalian cells or insect cells. So the very first step for purification is to perform cell lysis. So you need to rupture the cells to release the viral vector. In the lab scale, traditionally people use a freeze-thaw method, which is a very tedious method and almost impossible to scale up. Chemical method is actually efficient and scalable method, but we have discussed the concerns for using chemicals before, especially a very popular detergent, Triton X100, has already been banned in EU, which leaves the mechanical method is an attractive method for performing cell ruptures. But however, like I mentioned, when you choose your technology, you want not only very efficient cell rupture, but also minimize the damage and to achieve maximum yield. So the data showed here is through our collaboration with Corning. So in this study, AAVs were produced in Corning's latest Ascent FBR system and were harvested using the Microfluidizer processor. And as you can see from the graph to the right there, the Microfluidizer system was successfully scaled up with different cell concentrations up to 50 million cells per mil, which was the highest amount of cell concentration we tested. Microscope images here showed you the cells were nearly completely ruptured at all concentrations. Vector extraction was also comparable to other technologies, although the qPCR titer analysis, which showed on the top right there, suggested a slightly lower vector extraction compared to chemical lysis. However, the Western blot analysis showed on the bottom there actually indicate comparable results or even slightly better yield. I know there's some further investigation needs to be done in the vector analysis, but this study at least showed the proof concept that the Microfluidizer technology is an ideal solution for scalable, detergent-free process for manufacturing AAV vectors. Before I conclude my presentation, I just want to quickly show you our diverse product lines. And we have a select range of lab units, which is showed on the top. And we have a pilot and production system showed at the bottom. And as I mentioned, our strengths in cGMP design. So for all the pilot and production system, including one lab model. We also offer biopharma version designs, which are in compliance with cGMP. And with that, I'd like to thank everyone again for your attention, and be happy to answer any questions. If you haven't, we have a booth here, number 6. Make sure you stop by and talk to me or to our colleague. Thank you. Yeah, there's a few different ways. Yes, there are a few parameters, especially pressure, that needs to be adjusted. I didn't mention we also have different chamber designs, which has the different dimensions for the microchannel. To combine the pressure and the microchannel design, we were able to achieve different shear forces. So we have some, based on our experience, we have some good ideas. If you based on your formulation, we'll have some good starting point for you, but obviously there will be some optimization needs to be done. We also we have an applications lab in our office here. We have all of our machines, and we offer like proof of concept tests, and can work with you before even you consider using our equipment. Yeah.
Thank you again.
Thank you.